Okay, right, so welcome to this next video in which we are discussing G-protein coupled receptors. Okay, so in this video what we want to do is talk more about the concept of biased agonism. Okay, which is a concept that we've seen in previous videos, but I now want to give you some nice examples of where it's actually being used in clinical scenarios. Okay, right, so let me just summarize where we've got to with our G-protein coupled receptor signaling, and then we'll see what biased agonism is once again. Okay, so basically we have seen how G-protein coupled receptors can bind their ligands. Okay, these ligands can cause conformational changes in the G-protein coupled receptor, and then the active state G-protein coupled receptor is then capable of signaling through two different means now. Okay, one is that it's capable of activating certain heterotrimeric G proteins, and we've seen that each G-protein coupled receptor has certain heterotrimeric G proteins that it prefers to activate. Okay, but the other major way that G-protein coupled receptors can now signal is that they can be phosphorylated by G-protein coupled receptor kinase enzymes. Arrestin proteins can bind, and then that arrestin protein can have all sorts of other signaling cascades. Okay, now different G-protein coupled receptors will have differing abilities to activate heterotrimeric G proteins versus the arrestin signaling. Okay, so different G protein coupled receptors will have more of a tendency to do one or the other, basically. Okay, so not all G protein coupled receptors activate the arrestin signaling as much as each other, basically. Okay. Now, the classical view of G protein coupled receptor signaling was that all ligands, if they're agonists, induce the same conformational changes in G-protein coupled receptors, that there were only two states that a G-protein coupled receptor could be in, the inactive state and the active state, and that an agonist molecule merely moved the dynamic equilibrium towards the active state. That view is outdated. Okay, it's laughably oversimple. Okay, what we are becoming aware of now is that different ligands can cause different conformational changes in the same G-protein coupled receptor, and that these differing conformational changes have functional significance, i.e. they determine which heterotrimeric G proteins the G-protein coupled receptor can activate, and also how much of the arrestin signaling is it going to activate, basically. What's its tendency to activate the arrestin signaling and be desensitized? Okay, this is the concept of biased agonism, that different ligands can bias a G-protein coupled receptor to either signal through heterotrimeric G proteins or the arrestin signaling, and then also can bias G-protein coupled receptors to certain heterotrimeric G proteins over others. Okay, so I want to give you some examples of where this is actually clinically useful. Okay, so the drug Carvedilol, okay, with regards to the beta-1 receptor, we're going to study it. Okay, the beta-1 adrenergic receptor. Okay, right, so Carvedilol is a drug that is used to treat heart failure, and I'll tell you in a moment a little bit about heart failure and why Carvedilol is a nice idea for heart failure. Okay, but first let me tell you that Carvedilol is classically viewed as being a beta-1 antagonist. Okay, this is the little name for it that you will see if you read most pharmacology textbooks. You will see that it's a beta-1 antagonist. It is supposed to bind to the beta-1 receptor and not alter the signaling of the beta-1 receptor at all. Okay, it's supposed merely to block noradrenaline from being able to bind to the receptor and stimulate the receptor. Okay, that's the classical view of how the beta-1, how Carvedilol interacts with the beta-1 adrenergic receptor. Okay, however, it's not true. In actual fact, Carvedilol is a biased agonist. Okay, and we'll see how this is useful in just a moment. Okay, so we'll come back to this. What I now want to do is tell you a little bit about heart failure and what happens in heart failure. Okay, so there are loads of different ways that you can get heart failure. Firstly, I'll just define what heart failure is. Okay, so heart failure is when the heart fails the rest of the body. The heart becomes so weak that it is no longer capable of pumping adequate blood around the body to meet the needs of the peripheral tissue. Okay, so the heart's job in the body is to pump blood around the body, okay, to supply oxygen and nutrients to the peripheral tissues. When the heart becomes too weak to actually do that job, then there isn't enough blood going to the peripheral tissues. That is when the heart is failing the rest of the body, and that's what is meant by heart failure. Okay, and it kills you in the end because you're getting too little blood to your peripheral tissues. Okay, now, as I say, there are many different things that can lead to heart failure, but one of the common things that leads to heart failure is chronic hypertension, which is why we're also worried about our blood pressure. Okay, so if you have chronically high arterial blood pressure, this can lead to heart failure, and let me explain why. Okay, so if you have really high arterial blood pressure, so let's say your aortic blood pressure is very, very high, what does this mean for the heart? Well, remember, the heart has to pump blood into the aorta. If I draw a very simple picture of this, here is the heart shown as a box, okay? And basically the heart has to pump blood into the aorta here, okay? Like so, and then of course the blood will come back through little arterioles here, capillary beds, so I'll just draw a simple picture of the cardiovascular system. Then you'll have the venules here, and then the venous system will bring the blood back to the heart here. Okay, and I'm not showing the pulmonary circulation, that's included in all of this basically. Okay, so here's the heart. So basically, the heart has to pump blood into the arterial system here. So if you've got a massive, great arterial blood pressure, it's going to make it much more difficult for the heart to pump the blood into the aorta, okay? Because if the blood pressure here is really high, then the heart's going to have to force more blood into that blood vessel, and that's going to be really difficult if the pressure's really high here. Okay, so the heart starts to struggle, and in fact, the amount of blood it actually can pump into this really hypertensive arterial system is going to go down. So the cardiac output, which means how much blood the heart moves from the venous side into the arterial side in a minute, is going to fall. So CO is just short for cardiac output. Okay, now, when the cardiac output falls, that causes the activation of the sympathetic nervous system. Okay, so this is going to lead to the activation of the sympathetic nervous system. And the sympathetic nervous system innervates the heart. Okay, it releases noradrenaline onto the heart via cardiac nerves that innervate the heart. And the noradrenaline works on beta-1 adrenergic receptors on the surface of the cardiomyocytes, okay, which I'll draw here. So here is our classic structure for a G protein coupled receptor, and this is specifically now a beta-1 adrenergic receptor. Okay, now, the stimulation of beta-1 adrenergic receptors on the cardiomyocytes has two effects. Firstly, by acting on cardiomyocytes in the sinoatrial node, it causes the discharge rate to go up, which then causes heart rate to go up, HR to go up, so the heart beats now faster, okay? And also, it causes the contractility of all the cardiomyocytes to go up. So by working on all the other cardiomyocytes, it's going to cause the contractility to go up. Okay, so these are the two major effects of the sympathetic nervous system on the heart. The heart increases the rate at which it contracts. Okay, so it's going to beat more times, it increases the number of times it beats per minute, the heart rate. And also, by acting on all the cardiomyocytes, you make all of them contract harder. Okay, and the idea here is that this is going to bring cardiac output back up. So even though you've got the chronic hypertension, which is meaning that the heart really is struggling to eject blood into the arterial system, now, because we've increased the contractility of the cardiomyocytes, we've made them contract harder, okay, so that they generate more force and they can expel the blood out, basically, despite the really high blood pressure. Okay, we've also made them contract more often, so beating more often. That's going to help to bring back up the cardiac output, how much blood the heart moves. from the venous side to the arterial side in a minute. Okay, so that gets over the problem. The problem now is that chronic sympathetic stimulation of the heart in this way is not good. Okay, if you have the sympathetic nervous system activated temporarily, that's fine. Okay, that's part of the fight or flight response. It activates temporarily, the heart rate goes up, the contractility goes up. The idea is to deliver more blood to the skeletal muscle so that you can get yourself out or prepare for fighting. Okay, but now we've got it activated chronically, and that actually causes problems for the heart cells. Okay, in fact, chronic sympathetic stimulation in this way, so I'll put chronic SNS stimulation, leads to a phenomenon known as hypertrophy of the cardiomyocytes. Okay, so let me just explain what hypertrophy of the cardiomyocytes means. So basically, if you want to grow a piece of tissue, there are two ways that you could do it. So let's say we've got a piece of tissue here, and these are the cells of the tissue. If you want to make this tissue bigger, there are two ways that you can do it. Okay, either you can actually make the individual cells of the tissue bigger, like so. Okay, that kind of growth is known as hypertrophy. Okay, or hypertrophic growth. Okay, alternatively, you can just make the cells divide. Okay, so we could go from having 4 cells to having 9 cells, and that would also cause the tissue to grow. Okay, this sort of growth of a tissue is called hyperplasia. Okay, heart muscle cells, cardiomyocytes, are not capable of hyperplasia. Okay, they are not capable of dividing. Okay, they have very strict controls in place that pretty much mean that they are not capable of dividing at all. Okay, that's really one of the main reasons that you never ever hear about heart cancer. Okay, cancer of the heart. The reason that cancer very rarely, I mean it's not unheard of, okay, but very rarely ever develops in the heart is because the cardiomyocytes just have very powerful brakes on division, so it's difficult for you to get enough mutations in a cardiomyocyte that mean that the cardiomyocyte is going to end up over-dividing, which is the characteristic feature of cancer. Okay, so cardiomyocytes cannot divide, and in fact the cardiomyocytes that you have in your heart now are the same cells as you had when you were born. Okay, so you keep the same cardiomyocytes throughout your entire life. Okay, so if you want the heart to grow, there is only one way to do that, okay, and it's hypertrophy, making each individual cell of the heart bigger. And basically, chronic sympathetic nervous system stimulation of the heart can cause this. It causes the cardiomyocytes to become bigger. Okay, so let me draw this. So this might be one cardiomyocyte here, and now with chronic sympathetic stimulation, what can happen is its width can go up and its length can go up, okay? And this is the hypertrophied cardiomyocyte. So the heart ends up getting bigger, basically. Okay, now, the problem is that this kind of hypertrophy is what we would call pathological cardiac hypertrophy. Okay, things go horribly wrong, basically. What this seems to be is it seems to be a attempt by nature to actually make up for the problem that it has here. The problem is it doesn't seem as though nature has actually got the opportunity yet to perfect this hypertrophy. Okay, and it ends up going horribly wrong. So this is going to be called pathological hypertrophy. So what ends up happening is when the cardiomyocytes undergo this pathological hypertrophy, they get bigger like this. Temporarily, what this actually means is that this bigger cardiomyocyte is stronger. Okay, it's better at actually contracting. Okay, it generates more force. Fantastic, that's exactly what we need, because remember the whole problem here was that we had the chronic hypertension and that the heart was struggling to push blood into this hypertensive arterial system. Okay, so making the heart stronger seems like a brilliant idea. The problem is it's short-lived, okay? These become stronger for a while and then they just gradually lose their ability to contract completely. Okay, things start going horribly wrong for these poor cardiomyocytes. Okay, and it's thought that what really goes wrong in these cardiomyocytes is calcium signaling. Okay, so remember, for the cardiomyocyte to contract, the action potential that propagates along the membrane of the cardiomyocyte has to be converted into a calcium signal in the cytoplasm, which then causes the cardiomyocyte to contract. Basically, you get horrendous problems with the calcium signaling, the coupling of the action potential to the calcium signaling in these hypertrophy cardiomyocytes, and they just lose the ability to couple the action potential to the contraction. Okay, so gradually what ends up happening is the heart becomes weaker and weaker and weaker, okay, and this gets worse and worse and worse until the cardiac output descends to a level that it's no longer capable of sustaining the needs of the peripheral tissue, and then we would say that you're in heart failure. So hopefully you can appreciate how this is a chronic, horrendous cycle, because the more hypertrophy that occurs, the weaker the heart gets, the more the cardiac output goes up, the bigger the sympathetic nervous stimulation gets, and that's what drives the hypertrophy. So it's a vicious cycle of more cardiomyocytes of the heart undergoing this hypertrophy process, the heart getting bigger and bigger but weaker and weaker, basically, and that's how you descend into heart failure with chronic hypertension. Okay, so that's why doctors worry about chronic hypertension and what it can cause. Okay, heart failure is one of the major things it can lead to. It can also lead to other things such as atherosclerosis and all of the many complications that occur because of atherosclerosis. Okay, right, so Carvedilol then is used to treat heart failure, and how does it work? Well, the classic view of how it works is that it is a beta-1 antagonist. It binds to the beta-1 adrenergic receptor, doesn't stimulate the beta-1 adrenergic receptor, but blocks the noradrenaline from stimulating it, and therefore stops the chronic sympathetic nervous stimulation of the heart, and stops this vicious cycle, basically, stops more pathological hypertrophy from occurring, and stops the heart descending into such weakness that you go into heart failure. Okay, however, it seems that Carvedilol doesn't actually do that. It does more than that. In fact, it's better than that. The reason it's good for heart failure is better than just being a beta-1 antagonist. It is actually a biased agonist. Okay, so let me talk a little bit more about how the beta-1 adrenergic receptor causes the rise in heart rate and the rise in contractility. So basically, the beta-1 adrenergic receptor has two options for how it signals. Okay, one, it can signal through heterotrimeric G proteins, and the heterotrimeric G proteins it specifically activates are GS heterotrimeric G proteins, and it's the activation of the GS heterotrimeric G proteins which increases cyclic AMP and activates protein kinase A that causes the heart rate to go up and the contractility to go up. Okay. However, beta-1 adrenergic receptors can also activate arrestin. Okay, and the arrestin can then lead to the desensitization of the cardiomyocytes to noradrenaline, the removal of the beta-1 adrenergic receptors from the cell membrane of the cardiomyocytes. Okay, so the ideal drug here would be one that stops the GS coupling, because the chronic high heart rate and the chronic high contractility is what seems to drive the pathological hypertrophy. Okay, so the ideal drug would be one that stops the GS cascade but does activate the arrestin cascade and causes the desensitization. And that, we think, is what Carvedilol does. Carvedilol binds to the beta-1 adrenergic receptor, changes its conformation in such a clever way that now this G protein coupled receptor activates the arrestin, activates desensitization of the beta-1 adrenergic receptor, but doesn't activate the heterotrimeric G protein GS, okay, and therefore doesn't cause the chronically high heart rate and the chronic high contractility, which is what drives the cardiomyocytes into this pathological hypertrophy. Okay, right, so that's believed to be how Carvedilol works to treat heart failure. So it's a nice clinically used example of a biased agonist, and that's why it's so effective as a treatment for heart failure and stopping this pathological hypertrophy from progressing anymore. Okay, so I want to give one more example, okay, which is completely different to this. This was the beta-1 adrenergic receptor. Now what we're going to talk about is agonists, or rather biased agonists, at the mu-opioid receptor. Okay, so I'm going to give two examples of these drugs. Okay, so there is a drug called herkinorin, okay, and another drug called TRV-130, okay, which are now used clinically as painkillers, okay, analgesic drugs. And the way they work is very, very similar to morphine, okay? In fact, it's exactly the same as morphine. They activate mu-opioid receptors. However, they don't cause the tolerance that morphine causes. So morphine is an absolutely fantastic painkiller, and the way that it works is by activating the mu-opioid receptor. Okay, so this stands for mu, and then OR is opioid receptor, a receptor that we haven't mentioned before, but I should just say it is an example of a rhodopsin family G protein coupled receptor, and indeed all the opioid receptors are examples of G protein coupled receptors. Okay, so morphine produces its analgesic effect by activating mu-opioid receptors, and I'll show you in a moment one of the key sites where the mu-opioid receptors work. Okay, the problem is that morphine causes tolerance. Okay, it causes gradual reduction in how effective it is. Okay, so this is the concept of tolerance. The concept of tolerance is that how effective the drug becomes less as you use it more. So the more you take the drug, the less it works, basically. You become tolerant to the drug, and then you have to take higher doses to actually get the same effect as you got before. Okay, and that's not good. We don't like that in drugs. Okay, and this is what then leads to the problems when you come off morphine. It leads to the physical withdrawal symptoms that you get. Okay, right, so we would like, ideally, a drug which activates the analgesic effects but doesn't cause the tolerance. And that is what herkinorin and TRV-130 are, by being biased agonists at the mu-opioid receptor. Okay, so let me tell you a little bit about the mu-opioid receptor. Okay, so the mu-opioid receptor here, it couples to GI heterotrimeric G proteins. So here's the mu-opioid receptor, and it can activate GI heterotrimeric G proteins. So heterotrimeric G proteins where the alpha subunit is one of the I's. Okay, but it can also activate the arrestin signaling. Okay, and the arrestin can then lead to desensitization, the removal of the mu-opioid receptors from the plasma membrane of the cell, and it's this that then leads to the tolerance. The removal of the mu-opioid receptors leads to the tolerance to morphine, because if you're removing the mu-opioid receptors, then that's the target for morphine gone, and that's why the effect of morphine goes down as you continue to use it. So ideally, we would have a drug that just activates GI, because the GI is how it actually produces the pain-killing effect, and doesn't activate the arrestin here. Okay, so let me just give you some insight into how activation of the mu-opioid receptor helps relieve pain. So basically, the primary nociceptive afferent neuron, which carries the signal from a peripheral nociceptor, has its first synapse with a second-order neuron in the dorsal horn of the spinal cord. And I said that all rather quickly, so I'll say it again. So basically, let's just draw a little picture here. Let's say this is the skin here. Okay, so this is the epidermis and this is the dermis. Then, in the skin, you have the endings of sensory nerves that can have nociceptors on their ends. Okay, so a nociceptor is a sort of tree structure like this, okay, which has receptors in its membrane that can sense noxious stimuli. Okay, so the nociceptor means this terminal portion of the sensory neuron which has loads of receptors in it for noxious stimuli. Okay, and if some noxious stimulus is delivered, let's say intense pressure is delivered to the skin, then this can activate that nociceptor. Okay, and the nociceptor will then send action potentials up the axon here of this primary nociceptive afferent. Okay, like so. So this is the neuron with the nociceptor on its end. So this portion at the end of the neuron here, this was the nociceptor, this tree-like structure here, which has receptors for noxious stimuli within it. This generates the action potential in response to a noxious stimulus, and then the action potential will propagate up this axon here. Here is the cell body of this primary neuron here, okay? And this will be present in the dorsal root ganglion. And then this central process, so this process which went out to the nociceptor in the peripheral part of the body, this is called the peripheral process. And then the primary nociceptive afferent also has this central process which will then go into the spinal cord. Okay? And in the spinal cord, it will then synapse onto the second order neuron here, a dendritic spine of the second order neuron, which I'll have like this. So here is the cell body of the second order neuron, and it will then send its axon up the spinal cord to the brain, basically, in the spinothalamic tract. Now, one of the key sites, and I'm going to stress this, it's only one of the key sites that the mu-opioid receptors are present, and by which the agonists of mu-opioid receptors produce analgesia, is this first synapse here between the primary nociceptive afferent and the second order neuron here. So this is the secondary neuron. Okay, now, basically this synapse here, which I'll draw bigger like so, this is a glutamatergic synapse, okay? So the axon terminal of the primary nociceptive afferent is going to release the neurotransmitter glutamate onto the dendritic spine of our second order neuron here, okay? And that glutamate is then going to open ionotropic glutamate receptors such as the AMPA receptor, and that's going to allow in an excitatory postsynaptic current, an EPSC, and that is then going to contribute to whether this second order neuron fires an action potential. Okay, so all of the excitatory postsynaptic currents that this neuron is getting, along with the inhibitory postsynaptic currents, will all sum together to decide whether the second order neuron is going to fire an action potential. Basically, you have mu-opioid receptors present on the axon terminal of these presynaptic neurons and on the dendritic spine of these postsynaptic neurons here. Okay, and if you activate the mu-opioid receptors on the presynaptic neurons, it activates the GI heterotrimeric G protein and of course the arrestin signaling, but it's the GI that's important, and GI then leads to the amount of neurotransmitter that this axon terminal will release going down. Okay, so what I mean by that is every time the action potential fires, the axon terminal usually releases a certain amount of glutamate onto the postsynaptic cell here. After you've stimulated the mu-opioid receptor, the amount of neurotransmitter that will be released per action potential is going to go down. Okay? On the postsynaptic cell, the activation of the mu-opioid receptor leads to the cell letting in a smaller amount of excitatory postsynaptic current for a certain amount of glutamate. So if you release a certain amount of glutamate onto this cell, usually you get a certain amount of excitatory postsynaptic current. Once you've activated the mu-opioid receptor through the GI cascade, this will lead to a reduced excitatory postsynaptic current for that same amount of glutamate being released onto the postsynaptic cell. Okay, so you have reduced the amount of neurotransmitter being released, and you have reduced the response to the amount of neurotransmitter. Okay, so effectively you are reducing the ability of the primary nociceptive afferent to stimulate the secondary neuron here, which is called a spinothalamic projection neuron. Okay, because it projects from the spine to the thalamus. Okay, so this is often referred to as the gate theory of pain. Okay, because this is seen as the gate, basically, and the idea is that the stimulation of the mu-opioid receptors is closing the gate, it's closing the ability of these peripheral primary nociceptive afferents to stimulate the second order neurons and have an effect on the signaling to the brain. Okay, so that's how agonists of the mu-opioid receptors cause powerful analgesia by effectively closing this gate. You can fire the primary nociceptive afferent as much as you like because of the presence of the noxious stimuli, but if it can't actually stimulate the second-order neuron, then it's not going to actually have any effect on the conscious experience, okay? So it's not going to generate the conscious experience of pain. Okay, right, and that's how morphine is so effective as a painkiller. It binds to the mu-opioid receptor, it activates GI, and causes this closing of the gate. And the problem is that morphine also activates the arrestin, and that leads to the desensitization. It leads to it becoming less effective over time as these mu-opioid receptors will be removed. Herkinorin and TRV130 are biased agonists that favour the GI pathway over arrestin. Okay, so they cause conformational changes in the mu-opioid receptor that now favour the activation of GI but not arrestin, okay, so that they have the effect, the desired effect. They still produce this strong analgesia, but without activating the desensitization of the mu-opioid receptors, and therefore not causing the tolerance that morphine and other opiates cause. Okay, so that's a major, major advantage to these drugs, and they are now being clinically used as painkillers. Okay, right. So that now completes our discussion of biased agonism and indeed our discussion of G protein coupled receptors.